Well, welcome everyone to our March AMS virtual seminar. I'm Jonathan Plett. I am the president of the association, and it is my pleasure today to welcome our speaker, Eric, who has quite a broad background in working with a variety of fungal lifestyles. If you look into the publications that he has done, he's worked across AM fungi, pathogens like stripe rusts, orchid mycorrhiza as well. And he's also worked in many leading institutions around the world and has thus given him a broad understanding and basis within microbial ecology. So it is really fantastic to have him here today. Some of his early work was really close to my heart because he was working with Physolithis and how that promoted plant health, and then showing that how fragile this promotion can be when you have competition with other soil fungi present, showing that when some saprotrophs were there, Physolithis's benefit was lost. More recently, he has been working in publishing how endophytes are improving plant growth and tolerance, especially with abiotic conditions of different salt levels. So today, Eric is going to be talking about specifically about new technologies that we can be thinking about adapting within our research and how these new insights can give us a better understanding of plant microbe interactions. So with that, I will pass it over to you, Eric. Thank you.
Okay, perfect. So I will share some work that I did so far about the interaction with plants and microorganisms, especially with fungi. For this reason, the name of my presentation is about the interplay between plants and microorganisms. So to start my presentation, I would like to leave a talk from a movie from Interstellar. So what is happening is because, for example, in general, the Western societies, they perceive the ecosystem that is spread from the humans, and we saw the ecosystem only like a point of view of research that we can use for ourselves. And this is because influence from cultural, philosophical, and economic factors like religion, philosophy, schools, and industrialization, etc. But for example, we can learn from the Aboriginal perspective because they have a new point of view about the ecosystem. They say all of the elements, they are connected, all the humans are connected with nature, and we need to respect them. And for example, in this own language, they have like several words that refer to this connection. But this connection is more deeper than only plants, animals, and humans, because all the elements that belong to ecosystem, they are connected one with each other, and they are important for every step of the ecosystem sustainability. For example, the fungi. And for example, we can give you the example of the triple alliance between microorganisms, plants, and soil, because the three are connected and they need one of each other to shape the ecosystems. And as you know, the plants are not alone, because the plants can host a diverse community of microorganisms. So the plants can be called like a holobiont. For example, this microorganism can be found in the exosphere, can be found in the endosphere inside of the plant, and also in the rhizosphere. And this microorganism could be commensalists, could be parasites, and for example, sorry, out again. Sorry, and the plants can interact with different microorganisms at the same times, like for example with mycorrhiza or with parasites. So for this reason, it's important to explore beneficial microorganisms from the ecosystems, and to try to understand how beneficial can be for the plants and how we can use this beneficial microorganism to improve other plants, for example, the crops. And I can give you a work that we did with the plant Festuca rubus with species Purinosa. So this plant is a plant that we can find in the cliffs, like in the ocean Atlantic coast. And for example, one characteristic of these plants is they grow up in the crevices in the between rocks, and they are exposed from salinity from the sea coast or from the sea and from the strong winds. And this plant, it was known to be a natural host of the fungal endophyte Epicloy festucae that habits the above tissues of the plants, and the interaction between epicloy and these plants, they can produce alkaloids that could be toxic for insects like paramine or lolines, and also they can produce other alkaloids that could be toxic for mammals like lolitran and ergovaline. And so far, and until recently, the root microbiota, it was unknown. So from this work, I used plants from the north coast of Spain, and I tried to study the root microbiota and also the presence of epicloy. And something that I figured out is like more than 65% of the plants were were affected with epicloy festucae. So it's a very high, very high percentage. And also, I found a big variety of fungal endophytes in the roots. And it was interesting because I found a core microbiome, because from these 735 different species, it was presented more in 20% of the species. And I would like to highlighting the Fusarium oxyspore and diaport, because these two species I found in more than 50% of the plants. So, and this work, this result you can find in this paper from 2019. So with this high variety of endophytes, so I try to figure out how these endophytes can help the plant to support these stressful conditions. Because, for example, from the salinity stress. And for example, something that I figured out is this plant use the tissue tolerance mechanism to support the salinity conditions. So they keep the salt in the vacuoles, and for this they need to accumulate prolines in the cytoplasm. And for example, when I measure the concentration of proline in plants with salt and without salt, and for sure, I found a very big concentration of proline in plants with salt. But for example, when I study the salt concentration in plants in the presence of different microorganisms in fungal endophytes, I figured out the concentration of salt inside of the plants decrease. For example, I can give you the example of epicloy and Fusarium oxysporum. So it seems like in the presence of fungal endophytes, the plants also can adapt to this condition using a new mechanism. For example, the salt exclusion mechanism. For example, in some way, these endophytes help the plants to exclude, for example, to avoid the roots to absorb, to uptake the sodium. But at the same time, the plant can continue uptake water, for example. So, and these results also we can find in another paper from 2021. So I figured out these fungal endophytes can have like positive effect on the host plant. So I figured out if this plant, or I try to figure out if these fungal endophytes can have also a positive effect in no-host plants, for example, in crops. And for this way, I use, for example, the tomato plants. So in a work that I explore the effect, for example, the diaport, do you remember it was one of the most common endophytes in the festuca roots? And I used this fungal endophyte to study if can help the plant to support, for example, drought stress. And for example, for this research, I did 4 different treatments. It was plants with normal water without endophytes, plants with normal water with the diaport, and plants under drought stress without endophytes, and plants with, under drought stress with the diaport. And something interesting that I figured out, it was, for example, under drought stress, the biomass of plants decrease, but for example, in the presence of diaport, this decrease it was not like too expressive, for example, without diaport. So I try to figure out all these plants, all these fungal endophytes can help the plant to support the drought stress conditions. So something that I figured out is, for example, in the presence of diaport, they improve the photosynthesis of the plants. For example, we can see because the chlorophyll content will be higher in plants with diaport, and also under drought stress, plants with diaport will use the water more efficiently. Also, they will accumulate more proline in cytoplasm, and also they will produce more enzymatic antioxidants. So, and this work also you can find in another paper from 2023. So in this way, I figured out this diaport can help the tomato plants from example from abiotic stress like drought stress. So I try to figure out if you can be useful, for example, against a pathogen. In this case, I study against the Fusarium oxysporum form specialis lycopersicii. It's a pathogen from tomato plants. And something that I figured out is, for example, in the presence of, I studied the disease severities in a scale from 0 to 5. And for example, 0, it was plants without any symptoms from Fusarium, and 5, the plant, it was all destroyed and dead. And for sure, for example, in the absence of Fusarium, we don't find any damaged plant. And in presence of Fusarium, only with Fusarium, all the plants were destroyed. But interestingly, when in the presence of diaport, more than 50% of the plants, they are healthy. And only a few percent, like 40% of the plants, they present strong symptoms from Fusarium. So I try to figure out how this can be possible. So I study because, for example, what's happening with Fusarium? Fusarium comes from the soil. They enter in the plant from the roots, and they spread from the xylem, from the vascular tissues. And what I figured out, for example, the plant for sure only with Fusarium, all the plants and all the vessels, it was infected with Fusarium. But plants with diaport, some plants they don't present any damage plant, they don't present like vessel infected with Fusarium, and or in some parts only, like for example one vessel. And for example, in plants without diaport, the infection it was until the top, and for example with presence of and diaport, this infection it was only reach, for example, in the basis of the plants and never reach to the top. So in some way, the diaport like have an antagonist effect against Fusarium. So, and this work will be I submit this paper soon. So for sure will be approved, and more interesting result you can figure out in the paper. So But for example, all this work that we did, we use like normal and classics techniques. But for example, I think could be interesting to apply new technologies to study the interaction with plants and and microorganisms, for example, in fungi, and is what I did, for example, in my first postdoc in Ireland. So like we know, so a lot of process happens inside of the soil. And for example, and this process that happens inside of soil will be important for the development of the plants. But for example, it's important to try to understand what is happening there. And for example, when we are collecting plants, we are losing information because, for example, we are losing the architecture of the plants because, or we for example, we can collecting the plants, we are breaking the roots. So some information it was losing. So something that will be interesting to study is using some technology that we can study, for example, the roots in situ. So in this way, we apply the X-ray computer technology, and we can obtain X-ray image from the roots inside of the soil. So so I applied this project. I applied this technology in a project where I study the effect of herbivore mycorrhiza and potato cyst nematodes in tomato and potato plants. So I inoculate tomato and potato plants with mycorrhiza. I infected with potato cyst nematodes. So I did an analysis of the pots with the X-ray computer technique. Like it was like similar to, do you notice X-ray that we can find in hospital, but they change it to be used, for example, in this kind of situations. And with this technology, we can obtain 3D root image. And with this 3D root image, we can have like a full understanding about the root architecture. And all this could be possible. For example, the samples is fixed in a stage that we will be rotating 360 degrees, and the X-ray gun will take like a lot of different image. And after this image, we will use a software when we will put together, and we can obtain a 3D image. And for example, here we have several photographies from the same plants. For example, in the first one, we can see the potato plant inside of the soil. The soil will be in orange, and the plant will be in white. And we can see in different scale. For example, something we can do, we can work with it. For example, we can see all the pot, but for example, we can take the soil and we can see only the plant. And an interesting thing is, for example, we can obtain like a 3D image, like a video like this one, and we can have a full perspective of the root architecture. So what I did, for example, I'm giving you an example of work that we did. It was, for example, a full experiment that I inoculate. I use plants without any microorganism, plants only with mycorrhiza, and plants infected with only with potato cyst nematodes, and another plants inoculated and infected with both microorganisms, and I try to figure out if the presence of, in the absence of this microorganism, have like an impact of the root architecture. So and in below you can see like the views of 3D, like an example of one plant from each treatment. And for example, with this kind of technology, we can obtain the root volume. And for example, this is only some result that I obtained so far. But for example, with root volume that we verify, for example, in the presence of mycorrhiza, the volume increase in 4 weeks. when we start the treatment, independently the presence of cyst nematodes. And the same happens for the surface area of the roots. And also to be sure that it was I was doing everything correctly, I figured out if this root it was inoculated with the Herbacula mycorrhiza and we infected with potato cyst nematodes using microscopic analysis. But something that I figured out when I was doing this work, it was very hard to looking for cyst nematodes in soil because it's a very laborious work and we waste a lot of quantity of water and we lose a lot of time and it's very stressful. So something that I think about is, for example, I have soil with cyst nematodes. I have X-ray machine. Why I cannot use, for example, this machine to figure out or to try to identify cyst nematodes in soil? So it's what I did. So in the first stage, what I did is it was, for example, to take X-ray image from cyst nematodes to have an idea how the cyst nematodes looks like in a X-ray point of view. So for example, I use a cyst nematode and I take several pictures. And for example, I have a 2D image, and it was interesting because we can identify the different structure of the cyst, for example, the shell and the eggs inside. And after, with this image that I obtained from only cyst nematodes, I try to identify the cyst nematodes inside of the soil. So I start, for example, with a low quantity of soil, for example, with Eppendorf, and after I start to increase until like a 50 milliliters Falcon. And it was interesting because it was possible to identify cyst nematodes in the soil. And I use different kinds of soil to be sure that we can use this technology in different kind of soil. For example, only with sand and mixed with sand and soil. And all of the soil that I used, it was possible to identify the cyst. And for example, here we have a frontal perspective from a 2D image and a top perspective, and it's possible to identify, for example, like this round spheric thing that is the cyst, and the contrast with the soil. For example, something that we can figure out with the X-ray machine is, for example, in the presence of something constituted with carbon, will be less shiny than, for example, with minerals. And for example, we can also identify, for example, the air spore, because the air spore will be totally black. And for example, with this 3D image, not only we can identify the presence of the cyst nematodes, also we can identify in which part of the soil we can find the cyst nematodes. So in the first thing that I did, it was putting cyst nematodes in the soil, because for example, it was more easy to figure out. For example, I put 4 cyst nematodes in the Eppendorf, so I need to find 4 cyst nematodes in the X-ray machine. So, and in the last part, what I did, it was using soil with cyst nematodes, but I had no idea which kind of cyst, with the quantity of cyst nematodes that we can find this quantity of soil, and in the same way, it was possible to find the cyst nematodes. For example, here we have an example of a soil with a unknown quantity of cyst nematodes, and we can see perfectly the presence of cyst nematode. And also it was interesting because we can identify if the cyst nematode is full of eggs or if empty of eggs. And for example, here we have an example of a cyst nematode inside of soil full of eggs. And another thing that I did, it was, for example, to evaluate if the volume of the cyst nematode is correlated with the number of eggs. And it was interesting because, for example, bigger cyst nematodes with a bigger volume, they present much more eggs. And for example, another thing that I verify is, for example, empty cyst nematodes. The shell, it was like more, it was a little bit destroyed, and inside we can see like a lot of space, a lot of empty space. So recently I started my new project. I moved here to Canberra to start my new postdoc, like 1 month ago, 2 months ago, sorry. So currently I'm working the Schweinsinger group that I'm working in the genome biology of the Puccinus striformis tritici, that is a pathogen from wheat that I will study the genome, and I will try to study, or not, I don't try, I'm studying the virulence evolution of this pathogen. And also, I'm collaborating in a new project with the Professor Celeste Lin, that I'm working with genome from Australian orchid mycorrhiza, for example, Tulusnella serendipita, and because it will be the first time that we are doing something similar, and I hope soon I can have interesting results. So, in the end of my presentation, I would like to leave you with another thought from another movie. Yes, life always finds a way. And thank you for to be here. And I hope you enjoy my presentation. And if you have any question, you can contact me by my email, or you can connect me with Twitter, and make any question that you want. And also here, I'm open for any question. Thank you.
Fantastic. Thank you very much, Eric. That was a great overview of the research that you've been doing. A reminder to those in attendance, you can ask questions if you just pop it into the Q&A, click on the Q&A button that you have at the bottom there, and then we'll direct that to Eric. While people are thinking about questions, I have a couple of them. So with the diaporthe work that you were doing with Fusarium, so you found that the Fusarium was not in the vasculature anymore. Do you know why?
But you are talking about Fusarium in tomato, sorry?
In tomato.
Because
yep.
Something I forgot to mention in my presentation is the
And
Fusarium of fish port that.
Sorry, 2 seconds. You can stop sharing your screen now.
Okay.
Perfect. Then I don't have to watch myself twice.
Okay.
Yeah,
Okay,
so
sorry.
with the tomato work that you did, and you found that there was less Fusarium when you had the endophyte there. Do you know why that was the case?
I think the case, I cannot give you like a full response, because it's something that I'm trying to figure out what is happening there. But something that I can tell you, for example, Diaport is not using, for example, the phytohormones pathways in defense, because, for example, these plants with Diaport and Fusarium, the pathways of phytohormones, it was down-regulated by the presence of Fusarium, but the plant still wealthy. So it was interesting. So something that I think it was like a direct competition. For example, competition for space, because from both our organism is from soil. So they want to occupy some parts of the plant, like the niche of the plant. So they compete one each other for space inside of the plant.
Did you ever try and propagate Fusarium out of the roots as well to see if it actually had gotten into the roots?
Yeah, Fusarium is going into the roots. Because, for example, something that I figured out is, for example, I found some Fusarium in the roots, but they are not spread for all the plants. So in some way, the apport is pushing there down.
Great. Tom
And
May has, oh, yep.
so just because something that I forgot to mention in my presentation is the Fusarium spore that I found in Festuca is not the same Fusarium that I found that I used for tomato plants,
Okay,
because
great.
both they are Fusarium spore, but one is pathogen and another is endophyte.
Yep. Okay, great. Yeah, that's the fun part with oxysporum, isn't it? Um, Tom May has a question about the use of cartoon style drawings that you had. Were those yours or were you using someone else's? And are you planning on expanding using that method of communication in a broader audience?
No, I'm using this kind of cartoons because sometimes it's the best way to communicate for different kinds of people. Because,
Hmm.
for example, I don't know, for example, in the audience, they have students, but for example, if I'm talking with students, sometimes it's used to use like some language that I can approach to them and
Hmm.
to explain in the simple way about what I am doing and
Yeah.
try to be more interesting.
Yeah, no, definitely. They were great. A question from Salma. Hi, Eric. Nice presentation. When you work with X-ray potato cyst nematodes in the soil, is it also possible to use the same technology to identify mycorrhiza?
It's some, yes, I think it's something that I would like to try, but unfortunately, my scholarship or my fellowship in Ireland finished it before because the main part of this research it was about nematodes and not and the root architecture. So, for example, the identify cyst nematodes, it was something extra that I did. And yes, for sure, we can use this technology, for example, to identify other microorganisms, for example, to try to identify, for example, mycelium from roots.
Great. Also, Selma also has a follow-up question, is have you seen any phenotypic changes above ground when dealing with abiotic conditions like salinity and endophytes?
Yeah, sure. Because, for example, but we are talking about in Festuca.
I assume so.
Because, for example, in Festuca, no, I don't see any difference. What I see is, for example, when I apply, for example, in tomato.
Okay.
But for example, against salt in Festuca, no, everything because when you see different plants, you seem like similar. So the difference is more in like a biochemical way.
Yep. Cool. More comments about the cartoons. Teresa Labelle. So did you draw those cartoons yourself or were they borrowed from a paper or a book?
No, I did myself, but I'm using like artificial intelligence to help me with it.
Cool.
So I can write a code and ask
Yep.
here, I want this kind of cartoon with
Mhm.
this kind of elements.
Beautiful. No, really great. Teresa was wondering if you would be willing for others to borrow and attribute or borrow the use of them and attribute it to you.
Yeah, sure.
Cool. For the AM fungi, going back to the AM fungal story with the nematodes, so I was also kind of interested as well. It looked like from the scans that you had that there were more roots when the AM fungi were there. So I think you answered this question already, but did you try to quantify if the roots were growing faster or why the rooting was better when the AM fungi were there along with the nematodes?
No, for example, in the presence of with mycorrhiza, the roots growing faster. And for
They were or
example,
were not.
you can see
Sorry.
no,
They
they are
were?
growing faster.
Okay. Yep.
Yeah. Yes, because for example, something that we can figure out is, for example, we can find uh, a bigger number of roots in the presence of mycorrhiza, and they are growing faster. And for example, you can see the volume, and the volume will be higher in the presence of mycorrhiza, and for example, in this and the surface of roots will be higher. But for example, something I figure out is, for example, this and it was interesting because we can see this change in 2 weeks, and for example, also in 4 weeks. But in 4 weeks, they can decrease a little bit because it's not because the mycorrhiza is losing strength. It's more about space because I'm
Okay.
using pots, so the plants they don't have more space to continue growing.
Hmm.
But the effect of the mycorrhiza is there.
So if there were more roots, were there also more infections by the nematodes, or were the nematodes also decreasing at the same time?
Now nematodes decrease the number of
Okay.
roots.
No,
Yeah,
but when
but it's
in combination,
interesting.
yeah, in combination with the arbuscular mycorrhizal
No,
fungi, though, were they infecting the roots as frequently or no?
yes, no. The nematodes continue infecting the roots,
Hmm.
but they don't cause damage. Like for example, in the absence of mycorrhiza,
Hmm.
because for example, in some way, nematodes are taking resource from the plants, but mycorrhiza they are recovering this loss from the nematodes from itself, like giving resource to the plant.
Really cool.
Yes,
Well, I think that is the end of the questions that I have in the Q&A. Thank you very much, Eric, for joining us today. Thank you for that great presentation. I wish you all the best down at ANU, and we look
thank you
forward
so
to
much.
hearing more from you.
Yeah, sure. It's a pleasure to give this seminar. And
Thank you, everyone who was here in attendance, and for everyone who will be watching this later on YouTube. A reminder, we are here monthly, and so we will see everyone again next month.